Hello, Bio221 scholars. We will kick off Module 4 with an overview of the plasma membrane. Now, you'll remember that the plasma membrane determines cell shape. We can see here, this plasma membrane is here in this animal cell in purple surrounding the cell. We see the extracellular side, which is in the dark purple, and the intracellular side, which is in the light purple. So the plasma membrane determines cell shape, provides a border to protect the cell from the outside or extracellular environment, and it determines the nature of the cell's interaction with its environment. So we're going to look closely at just a little slice of this plasma membrane as we move through the module, really. The plasma membrane has a unique property called selective permeability. And being selectively permeable means that cells can exclude some substances, take in others, and excrete things in a controlled manner. And we will look at this as we move through the module through active transport, passive transport, and bulk transport. The plasma membrane carries markers that allow the cells to recognize one another, and we can see them as these glycoproteins here. This is vital for tissue and organ formation during early development and later plays a role in helping cells of the immune system identify other things in the body as self and non-self. Another important factor or important property of the plasma membrane is something we call the fluid mosaic model. The plasma membrane must be very flexible to allow certain cells to change shape and size as they pass through narrow capillaries. So that's the spaces in between these phospholipids. So the cell must be able to adjust, the plasma membrane must be able to adjust to allow these items to pass through and to communicate with other cells. So let's just go quickly through the principal components of the plasma membrane. Take some preliminary notes here, and then you can add to them as you go through the other mini lectures and activities of the module. So the principal components of a plasma membrane are lipids. So we can see the famous phospholipid bilayer here, but then also these yellow structures here are cholesterols, which we remember from module 2, are also lipids. There's some proteins. That's all the things in blue that we can see here. Some proteins have carbohydrates attached to them, like these glycoproteins on the outside. These lipids have a carbohydrate, so they would be glycolipids. So you can see that glyco means carbohydrate. And all right, and so then there's so those are the lipids, proteins, carbohydrates, and then phospholipids we've talked about a little bit, but they're really important. So a phospholipid is a molecule that consists of glycerol, which is here, the orange head, so a glycerol head, and these two fatty acid tails. So the head is hydrophilic and polar, meaning that it likes water, and the tails are hydrophobic and nonpolar, meaning they do not like water, they repel water. Cholesterol, these yellow pieces that we see here embedded in the phospholipid membrane, are another lipid, and they're found alongside the phospholipids in the core of the membrane. So cholesterols are embedded on the inside, and they provide structure and rigidity to the cell membrane. And then there's these proteins. Proteins are receivers of extracellular inputs and can be activators of intracellular processes, as we've talked about with enzyme-substrate interactions. Proteins can also provide extracellular attachment sites or work as receptors for hormones and growth factors and other things. And they also can activate intracellular response cascades when binding happens. We'll look at that a little bit later as well. Just a little note, sometimes receptors are hijacked by viruses like COVID-19 and HIV. This allows these viruses to gain entry into the cells, and then at times the genes encoding the receptors on the outside of the plasma membrane can become mutated. So instead of waiting for the substrate, then they can be activated automatically, kind of in a never-ending loop, causing the process of signal transduction to malfunction, which can have disastrous consequences. And we're seeing this is exactly what's happening with COVID-19 right now. All right, so there's a few types of proteins that I just want to point out, a couple. There are integral proteins, which are these that span, they span the plasma membrane. So there's parts visible in the extracellular space, in the intracellular space, and then in the intracellular matrix, which is that space in between both sides. Integral proteins are complex, and usually they're receptors. They can communicate, because they span the entire plasma membrane, they can communicate from the intracellular to the extracellular environment. Then there's peripheral proteins, like some of these here that we can see do not span. They exist only on one side, and generally they provide structural attachments for the fibers of the cytoskeleton, which are these gray cords we see down here. And they can also act as cell recognition sites. These are sometimes referred to as cell-specific proteins. So the body recognizes its own proteins so it can attack foreign proteins associated with invasive pathogens. And these peripheral membrane proteins are responsible for those labels of foreign or self. Sometimes enzymes can be integral or peripheral. All right, and then so what about substrate? So what's binding to these peripheral integral proteins? Mostly, if we look at these glycoproteins here with the carbohydrate attached, these glycoproteins are always found on the exterior surface of the cell, so interacting with the extracellular environment. And they're bound either to other proteins like this glycoprotein or to lipids like this glycolipid. We will not see these strings of carbohydrates on their own. They can't link up with the phospholipids themselves. Okay, so this is an overview. So as you go through the module, we'll look at each of these components of the plasma membrane a little more closely and see how their structure relates to their function. And we'll also look at how the cell regulates what items are passing from the extracellular to the intracellular environment and vice versa. Okay, so now you can head on to the next activity.